right. So my name is Sara Adar. I'm a professor of epidemiology at the University of Michigan School of Public Health, and I am also a member of the HEI Review Committee. I am here along with David Dorman, who is on the HEI Research Committee, co-hosting this. And on behalf of the two of us, as well as Eva and Anna, who are at HEI and really put this work, this symposium together, we wanted to welcome you to this symposium on Smog to Synapse. In effect, what we're going to be talking about today is really how does air pollution impact our brains? I don't think I have to tell anyone of the importance of the brain, right? It is our control center for our bodies. It is our source of thought, is our source of, personality and being. So as you might imagine, if there's disruptions to the healthy activity of the brain, right, the burdens can be quite large. I suspect, given that 1 out of 3 of us in our lifetimes will experience a neurologic condition, my guess is everyone in the room has a loved one, a parent, a grandparent, something like this that you've watched experience dementia, maybe Parkinson's. And so personally, right, the burden's not only on that person, but also on the family and on the medical system. So it should not be a massive surprise that the Lancet Global Burden of Disease study has listed neurological conditions as a #1 cause of global burden of disease. I put up this slide recognizing that it's been real hard to see the details from the back, so I'll walk you through it. This is also from the Lancet, and what I like about this slide, it's showing you the disability-adjusted life years on the y-axis. It's broken down. The bars are by age group, and the colors are different disease types. Males on the left, females on the right. It's showing you, right, that different diseases are impacting people at different periods of life. It's not surprising. Infectious disease origins of, meningitis, encephalitis are what's driving those pink colors for childhood. As we start to progress into midlife and into older ages, the dark blue is for stroke, the pale blue is dementia, and those beautiful teal colors represent Parkinson's, right? So showing you that we have this shift in the type of brain health issues that arise by time in the life course. So I use that when I am thinking about what are the causes of disease that we can intervene on, how do we improve health? It really raises to me a couple of points. One is that we have to be thinking about this as a life course issue. So we want to be thinking about, are there critical times that we have exposures that matter more than others? Are they interplaying off of each other? And also, again, this idea of, synergy or antagonism across different factors. So when things occur together, does that actually help us or hurt us? So, HEI has been doing amazing work in this area related to air pollution, which is one of the risk factors, the risk factor we're talking a lot about today. In looking back, right, some of the first work was in 2017, looking at neurodegeneration in older adults. And now, right, it's all the way through today. We have a great research project that's ongoing by Dr. Carlston and Cleland, looking at wildfire smoke in the brain. Super interesting. Poster is here. Everybody should take a look at it. Hopefully the topics today will inspire you. If you'd like to see any of these reports, they are all online, and you can learn a bit more about them. Okay, in my last slide for us, I'm going to pause for one really brief advertisement, and hopefully it doesn't come across too much as like a self-serving thing here, but I wanted to just raise your attention to a new project that's ongoing. Actually, a lot of people in this community have been contributing to this effort. It's called the Gateway Exposome Coordinating Center. And the reason I bring it up is because it's an infrastructure project where we are working with funds from the National Institute of Aging to both help scientists and the community identify the top research priorities, create publicly available free data and guidance and training opportunities for people to get into this area of brain health and risk factors for that more easily. So that was just, pardon me, a quick little plug on that. And now it is the wonderful time to introduce our speakers. We have three great researchers lined up for you. First, we have Berchan Ikiz, who's going to be giving us a beautiful overview talk about this, air pollution and environment and the brain. Dr. Jennifer Weave is going to give a wonderful talk about the epidemiologic research and evidence that we have specifically related to dementia. And then finally, last but definitely not least, we have a great talk from Dr. Deborah Corey-Slecha, who's going to be talking about the toxicology literature in this space. So without further ado, Berchan.
Good morning, everyone. Let me see. Well, good morning. Thank you so much, Sara, for your kind introduction and for setting the stage so perfectly. And thank you to HEI for inviting me to be here today. This is my first time at HEI, and I had the privilege to sit next to John Samet at the dinner last night. And I was like, I don't think this is your first time at an HEI meeting. So he gave me a whole overview of the long and impactful history of HEI, and it was wonderful. So thank you for having me here. It's a true honor as a neuroscientist to be here to talk to you about what we know in the state of science of neurodegenerative diseases and environmental factors and air pollution. When I started my career as a PhD student 20 years ago, not to give away my age, our understanding when we went to study the neurobiology of mechanisms of neurodegenerative diseases, we thought, okay, we have to look inside our brains. Let's look at those neurons that are dying and try to understand what's happening with these diseases. And over the years, we started understanding that was a very limited way of thinking, because those neurons that die during the disease process are living in an ecosystem with other neurons and non-neuronal cells, such as glial cells, microglia, astrocytes, that actually help them shape their well-being and health. And our brains don't live in isolation. They're connected to the rest of our bodies. We're learning more and more now that even Parkinson's disease may be starting in the enteric neurons in our gut and progress through the gut-brain axis into our brains. And we know that kidneys have connection to our cognition. And so when we talk about healthy brains, we have to also talk about healthy bodies. And our bodies live in an environment, a natural environment, a built environment, and a social environment, and all of those are interconnected. So today I want to give you an overview of what we know about the environmental factors. I promise I'll be mostly talking about air pollution and neurodegenerative diseases. So Sara shared this image earlier in her talk, talking about the global burden of neurological diseases. Why do we care? Why are we here talking about the brain this morning? And it is true that neurodegenerative diseases are now considered one of the major, highest global burden of diseases in our lifetimes, and especially late stages of life. And what we are learning, And today, about 50 million people globally are living with dementia. About 10 to 12 million people are living with Parkinson's disease. Another 5 million are living with other forms of neurodegenerative diseases, Huntington's disease, amyotrophic lateral sclerosis. And these numbers are expected to increase to an addition of 10 million people annually joining this population, with the estimates that, and please remember this number because I'm going to quiz you now, but I'm going to bring it back again. These numbers are expected to triple by 2050. And so huge, costing trillions of dollars in our economies, but also, as Sara had mentioned, a huge cost on our communities, on our families. And what we're learning is genetics only explains very little. It increases the vulnerability to having a neurodegenerative disease, but doesn't tell the whole picture. In fact, only about 10% of the cases are caused by genetic factors. For the rest of it is really impacted by the way we live our lives, the lifestyle factors, and also where we live, the environmental factors. And up to 45% of dementia risk is considered modifiable risk factors. The diet we have, how we manage our hypertension or vision loss or hearing loss or even depression and anxiety. These all play a role in leading our dementia risk and other neurodegenerative disease risk as well. And more the exposome studies we are doing that are coming up in the last 2-3 years is really on thousands of brain imaging samples and cerebrospinal fluid samples, and looking at environmental exposomes, social exposomes are giving us a better picture of how important environments are in shaping our brains and their aging, their resilience or vulnerability, and the neurodegenerative disease risk. And this is, so the air we breathe, how much green spaces we have, proximity to where we live, even the political climate of the country we live in. I don't know what it says about the United States, but something to think about, can have impact on our brains. And another really important thing we're understanding that Sara had mentioned is the importance of brain health across the lifespan. Even for neurodegenerative diseases are considered late stage diseases happening highest around 70 plus of age. But the disease may start years, sometimes decades earlier. And we're learning that it's really important, the way we live our life, the diet, exercise we do in midlife, the amount of education we get, or how we manage our mental health in youth, and even the neurodevelopment of children and fetuses can have impact on building our brain's resilience and managing our risk for neurodegenerative diseases. So these are some of the environmental factors we have the most data on the impacts of our brain health. As our climate is changing, we are experiencing more frequent and severe heat waves. Our world is warming up, and our brains function the best when they're in a certain environment and a certain temperature. And anything that deviates from that temperature, even if it's 1 degree Celsius, can cause stress on our brains, cause inflammatory responses, oxidative stress, misfiring of the neurons, and that kind of miscommunication leads to cognitive impairment, memory loss, learning loss for children, also worsening of symptoms if someone's experiencing neurological disorders such as multiple sclerosis, epilepsy, or psychiatric conditions like bipolar disorder, schizophrenia. Being exposed to heat can have real detrimental effects, even for short term, can have detrimental effects on our brain health. And we're also seeing more and more extreme weather events happening, wildfires, and floods and hurricanes that can really contaminate our water, our soil, our air. My family, we used to live in Los Angeles last year, and we experienced the wildfires. And not only the wildfires smoked, we learned carried all kinds of harmful particles. Even my child's preschool sandbox, we tested and found contamination of heavy metals. As these events are happening more and more, people are being exposed to these harmful particles that can really be harmful to neurodevelopment of children, increase the risk for stroke and dementia. And also these kind of events can introduce huge amounts of psychological stress among the impacted communities, increasing the risk for PTSD, depression, anxiety, and cognitive impairment even years after one acute event happens. One of my colleagues, Yoko Nomura at CUNY, had done a study about 15 years ago. Maybe some of you may remember the Superstorm Sandy that happened in the New Jersey, New York area. I used to live in New York at the time. Half of Manhattan lost its power. And she looked at the pregnant women who had experienced that storm quite seriously. They lost their homes, they had to move out for a while. And then she looked at these pregnant women's babies afterwards. And these babies had a higher risk of being born earlier and with a lower weight, which really affects their neurodevelopment. And these babies afterwards show higher risk for neurodevelopmental delays, for having been diagnosed with autism and ADHD, and in early youth having more mental health problems. My colleague Frederica Pereira had done long cohort studies over 20, 30 years looking at similar things in for air pollution in Harlem, New York, and Krakow, Poland, and observed very similar things. So even prenatal exposure to these pollutants, to these environmental factors, can have long-lasting intergenerational impacts on our brains. And we know the most data on air pollution among these environmental factors. Air pollution today is considered one of the major modifiable risk factors for neurodegenerative diseases. In 2019, the World Health Organization had come up with its first dementia prevention guidelines, focusing more on the lifestyle factors. And over the next 7 years at this point, had been revising the data and looking at how they could revise their guidelines, which is going to come out later this year, and they're including for the first time one environmental factor in those prevention guidelines, and that will be air pollution. And the State of Global Air report developed by HEI last year had found that in 2023, more than 1 out of 4 dementia deaths were attributable to air pollution, about 29%. That makes up 626,000 people, dementia deaths were attributable to air pollution, with the highest burden in lower and middle income countries, in Africa, and in Asia. So I want to come back to the beginning of the talk when I talked about, remember this number, I said don't forget the numbers are going to be expected to triple by 2050, the 50 million dementia cases. Well, those estimates were done looking at only one single risk factor, and that was old age. They thought, okay, because this is the trajectory of the aging population, we expect the numbers of people living with dementia and other neurodegenerative diseases to triple. But that did not include what we know about the environmental risk factors. With what we know with environmental risk factors, if we don't do anything, that tripled. The number of 3 was probably a huge underestimate, that we should be having a lot more people having these diseases, but not only that, but having them much earlier. I lead an international global network called Neuroclimate Working Group. We have physicians, researchers, policymakers working at the intersection of neuroscience and brain health and climate change. And we get clinicians coming from Macedonia, India, Pakistan, some of the cities that has the worst air quality. And they come join our group because in their day-to-day life, they are seeing more patients in their 50s, 60s coming to their clinic with memory problems, with cognitive impairment that looks like Alzheimer's, but they have been dismissed by all the other clinicians saying, it must be hormones, you're too young, it must be maybe depression, and they get tested and diagnosed with Alzheimer's without having any genetic marker that would cause them to have an early onset dementia. So these are becoming a scary trend, and so because the brain age is faster with these environmental factors. Oh, here we go. So how do the pollutants enter our brains and increase our risk for neurodegenerative diseases? Deborah is going to talk in detail about these, but there are 2 scenarios. One is that it may be entering our brains directly through the olfactory bulb, through inhalation, nasal inhalation, and the other is through systemic inflammation. As our lungs and other organs get inflamed from the particles in the air, they release inflammatory markers such as chemokines and cytokines, and sometimes the particles themselves get into the bloodstream. And we have a protect the blood-brain barrier, but it's not perfect. It gets leaky, especially in older age, in children, in people with existing conditions, and even extreme heat makes the blood-brain barrier more leaky, allowing for these inflammatory markers or harmful particles to enter the brain and cause more inflammation, oxidative stress, mitochondrial dysfunction, and eventual degeneration of the neurons. And we are all impacted by polluted air and harmful environmental factors, but obviously not equally. I don't have to say it for the audience here, but we know that children, pregnant women, fetuses, older adults, people with pre-existing conditions, and more importantly, people living in vulnerable places, low income, marginalized communities, the frontline communities, coming from lower and middle income countries where the exposures are the worst, but the protective measures are lowest, are the ones that are impacted the most. And we have to think about in our research work, in our prevention work, in protecting these communities. So those are some of the things we know, but there's still a lot of things we don't know. And I'm sure my colleagues Jennifer and Deborah will be talking more in detail about from the epidemiologist side and from mechanism side. One of the things Sara had mentioned we still don't know is the synergistic impacts of compounding factors such as heat and air pollution, or wildfire smoke and emotional trauma of having experienced an extreme weather event. What do they do together to our brains? And what we are in real life, in a lab environment, we may be able to isolate some of these factors and study one by one, but in reality, we are experiencing multiple of these factors all at the same time for long periods of time. And it's really important if we want to get a true picture of what's happening to our brains, it's important to be able to see them together. We also have an incomplete mechanistic understanding and a big equity gap in the researches we do, not looking enough to the populations and communities that are most impacted. And this is a perfect place to be discussing because this is exactly where we happen. We need to bring the health systems and science and policy all together in thinking about solutions as well. So I'm gonna invite you to take a second long break and take a deep breath with me. Good thing we have clean air here. And reshift our thinking a little bit, because what I tell, and you have been probably in the air pollution field longer than I have, these things can be really dark and scary to talk about to a lot of people. But in knowing this knowledge we have, I had a conversation with a colleague last night at the dinner where we talk about what do we do with the data we have, this data we have. It's really important to think about how can we turn it into, translate it into solutions. And the good thing we know is that just like nature, our brains are incredibly resilient and they are very plastic. And so they can rewire themselves and restore the damage that is done to them, given the opportunity. So in the actions we can do into cleaning our air, cleaning our environment, making our planet more livable and healthier, we can also support the brain health of our brains as well. And science shows us that's the case. In long cohort studies that have been done over 10 years, looking at thousands of women ages 75 to 92 years old in areas that had the air getting the cleanest across the US, they had found that the dementia risk had become 20% lower with slower cognitive decline and 2 to 3 years of younger slower cognitive aging. And these studies were replicated by longer 15-year-long Medicare studies done on Medicare patients, as well as in France and China, showing repeatedly that cleaner air led to lower dementia risk and healthier brains. A similar study was done in China in 2015-2016 when the clean air regulations passed and the air cleaned out. Within weeks, they were able to show suicide rates dropping by 30%, and they were able to causally infer it to the air getting cleaner. So there are huge co-benefits to our brains when we think about climate and clean air policies and redesigning our living spaces, whether it's our homes, whether it's our schools, cities, with the brain health in mind, in creating green spaces that can clean infrastructure, cleaning our air, water, soil, and making our places more resilient to heat and extreme weather events, and also creating places for social connection to happen. These are really beneficial for our brain health. And of course, carrying this knowledge and science and evidence into policy-relevant solutions, both at very local, citywide stages. We had heard from Sara and Christy yesterday in Chicago about what they're doing right here in the city with all those sensors and working with communities. There are a lot of things we can do locally in our cities, in our towns, and carry that action forward nationally and multilaterally with talking about the clean energy transitions, bringing brain health into conversations around healthcare policies, climate policies, clean air policies. What we cannot measure, we cannot change. So bringing the brain health outcome data and metrics into these conversations can really benefit in making our environments cleaner and better, and also our brains. So this is it from me. Thank you so much for your attention. If you would be interested in learning more about our Neuroclimate Working Group and our work, you can check out the QR code or the website neuroclimate.org. And these are some of the books written by our members and some of the articles also written by our group members. Thank you.
Hi, thank you for inviting me here. I have been tasked with telling you about what the epidemiologic evidence says about how air pollution might promote neurodegenerative pathology and etiology. I will be focusing on Alzheimer's disease, dementia, cognitive performance, and cognitive decline, not because other neurodegenerative conditions such as Parkinson's and ALS are not important, but because the evidence is most voluminous with respect to these outcomes. So dementia is a syndrome, and it's defined by deficits in memory and other cognitive functions that mark a decline from a previous level and are to such an extent that they impair a person's ability to perform everyday tasks. Now, Alzheimer's disease involves pathologies, neuropathologies, that contribute to the development of dementia. But I wanted to tell you that Alzheimer's disease pathology rarely occurs by itself. And what you're seeing here are the results of brain autopsies of 447 people, here in Chicago actually, who were all diagnosed with what we call clinically Alzheimer's disease dementia before they died. And there should be two things that stand out here. One, notice that second row, that Alzheimer's disease neuropathology is quite rare when it just occurring alone, I should say. It's actually really common, but by itself, it's quite rare. Far more common is Alzheimer's disease neuropathology in combination with other neuropathologies, including cerebral vascular pathologies. Now, the kinds of outcomes that we study with respect to air pollution and dementia, actually any exposure in dementia, include these. And I'm going to represent this with the hypothetical cognitive trajectories of 4 people. Many studies evaluate cognitive level in relation to air pollution. Some evaluate rate of cognitive decline, and others, obviously, look at the incidence of dementia in relation to exposure. Still others take advantage of brain MRI to evaluate neural atrophy, as well as cerebral vascular pathology. And with that, I want to just get to the state of the epidemiologic evidence on air pollution and dementia. And for that, I'm going to rely on 3 systematic reviews, so some may look familiar to you. And these are getting a little old, but the message remains the same. Let me run through those messages with you. First, I want to tell you about the US EPA Integrated Science Assessment for PM, which concluded that long-term exposure to PM 2.5 is, quote, likely to be causal in relation to, quote, nervous system effects. And by nervous system effects, a lot of that evidence had to do with dementia. That was a new sort of set of findings compared with the previous ISA. second review that my colleagues and I conducted a few years ago, where we focused on 35 studies of cognitive function, decline, dementia, and brain MRI that met quality review criteria. And the loudest signal in those data was an association between higher exposure to PM2.5 and rate of cognitive decline, so faster decline with more exposure. With respect to other exposure outcome pairs at the time, the evidence was mixed or sparse by comparison. And then finally, I want to tell you about Alyssa Wilker's review that appeared a couple of years ago, where they focused on 16 studies of air pollution in relation to dementia. And they found, in summary, an association overall of PM2.5 with higher dementia risk, although that was less strong in studies that used administrative records, such as medical records and claims. And with respect to other pollutants, the evidence was not quite as strong. And so you should be getting the message here. There's some common themes here, which is that PM2.5 appears to be the most consistently observed bad actor. It doesn't mean it's the only, but that is where the evidence is strongest. So with that, I want to turn to some recent developments in this line of inquiry, and first focus on developments on the outcome side, asking, does exposure to air pollution contribute to the neuropathology that drives the development of dementia? The emphasis has been on the pathology of Alzheimer's disease. So by that I mean beta amyloid plaques that build up between neurons and impair their ability to communicate, and hyperphosphorylated tau, which builds up within neurons and affects a neuron's ability to function. I'm oversimplifying, obviously. And so I'm going to talk a little bit about two studies that evaluated exposure to air pollutants in relation to biomarkers of these two pathologic signatures in cerebral spinal fluid. And first, I want to tell you about the Chinese longitudinal healthy longevity survey. There's a lot of long things in there. Is over 1,000 people. And what they found is that higher exposure to PM2.5 was associated with impaired clearance of beta amyloid from the brain. This is a biomarker that's consistent with that. We want to clear that beta amyloid so we don't build up plaques. They also found an association with a biomarker indicative of buildup of tau. They looked at nitrogen dioxide and ozone and did not report specific estimates, only that those associations were, quote, not statistically significant. Closer to home, just up north in Wisconsin, we have results from the Wisconsin Alzheimer's Disease Research Center and Wisconsin Registry for Alzheimer's Prevention. And in this study of nearly 800 people, they did not see much of an association of PM2.5 or ozone with the biomarker of clearance of beta amyloid from the brain. They did see adverse associations of these two pollutant exposures with buildup of tau. Now, before I go on here, I want to just point out, cerebral spinal fluid is awesome. It helps us a lot, and it's also it offers us a window to what is happening in the brain, but it's not specific to location. So, for example, let me just use tau as an example. The location of tau in our brains essentially drives the resulting manifestation of a clinical condition. So tau also occurs in chronic traumatic encephalopathy, the condition that happens in people who are involved in contact sports, but it's a different location. So this cannot tell us the difference between tau for CTE and tau for Alzheimer's. And what's interesting is that these folks in the Wisconsin study also underwent PET brain imaging of beta amyloid and tau, and with those data, they didn't really see much of association with those biomarkers in the brain. So we have autopsy studies, though, and this is an emerging sort of line of inquiry here. So most of those have been about PM2.5 exposure, and with autopsy studies, we can look at where these pathologies are in the brain. And I want to first give a shout out to a study based in the Adult Changes in Thought. This was led by Rachel Schaffer and involves decedents, I should say 800 plus decedents. And what she did is she estimated the exposure to PM2.5 in the 10 years prior to death. And what she found was higher exposure was associated with a measure of amyloid beta in the regions that are affected by Alzheimer's disease. This is called a CRAD score. But she didn't see much with respect to pathologic tau, as indicated by Brock's stage, nor did she see much in relation to a sort of composite measure of Alzheimer's pathology, indicated by the ABC score. At the University of Pennsylvania, investigators studied the brains of 602 people who had been diagnosed with Alzheimer's dementia or Parkinson's disease prior to death, and found positive associations of PM2.5 exposure in the year prior to death in relation to all three of these outcomes. And then at Emory, a study led by Christensen of 224 people living in the community looked at traffic-related PM2.5 in the year before death and found positive associations again with all three, but most notably the measure of beta amyloid buildup. Now, there are challenges with conducting autopsy studies in relation to the association of PM2.5 with Alzheimer's disease. And the first is selection bias. The people who agree to undergo autopsy and who die, and the age at which they die, these are all related to dementia risk. It's a pretty consistent phenomenon across studies. In general, people who are aware of their family risk and are higher risk are generally more likely to consent to autopsy. And because of this, it's really important that we ask whether exposure to air pollution is related to these processes as well. I want to credit Rachel Schaffer, the investigator for the Adult Changes in Thought study, who addressed this, at least in part, using a weighting technique. And so it's not commonly done, and it's actually really hard to do, but shout out to her for doing that. And it did affect her results and made them stronger. Her study's also marked by this feature of involving people living in the community as opposed to recruited from a clinical setting, and we know that that also can skew what we observe. Some other challenges, the exposure assessment. All of these studies anchor exposure to the time of death. Is the end of life the right period to be looking at? I mean, clearly something's showing up, but it's worth acknowledging that many people at the end of life live somewhere else than where they've been living for most of their lives. And then I want to mention outcome assessments. These are all focused on Alzheimer's disease pathology, neuropathology, and yet we know that this is not the only neuropathology that drives the manifestations of dementia symptoms. Flashback. A lot of those cases involve mixed pathology. And that brings me to vascular. As you've seen, Alzheimer's disease-only dementias are rare. And most people with dementia have some form of cerebral vascular pathology in their brains. And we know that having this pathology it seems to potentiate the presence of Alzheimer's pathology in terms of manifesting symptoms. In fact, I want to just highlight the results of the recently finished, what's actually still going, US Pointer trial. This is a lifestyle intervention trial to slow the rate of cognitive decline in people with mild cognitive impairment or at risk for dementia. So that intervention worked. Like, people's cognitive decline slowed. But the specific cognitive functions that decline were more consistent with what is affected by vascular processes than by amyloid and tau processes, so more executive function benefits than memory benefits. I'm preaching to the choir to tell you that air pollution has cardiovascular effects. But before you start thinking, oh, we should just start looking at vascular dementia, this condition is extremely hard to diagnose consistently. So let's turn back to autopsy studies, shall we? So I want to introduce you to the ARANOS study. This is a study that I'm leading here, based in Chicago, actually, and this involves people who were part of 4 cohort studies at the Rush Alzheimer's Disease Research Center, and specifically Dr. Tara Jensen, a postdoc working for me, just finished up a study where she estimated exposures to PM2.5, NO2, NOx, and ozone in the 5 years before everyone died. And she looked at these in relation to 12 neuropathologic outcomes. 3 AD, or Alzheimer's outcomes, 6 cerebral vascular outcomes, and 3 measures related to other pathologies that drive the development of dementia. And here's a snippet of what she found. What I'm showing you are the results in relation to cerebral arteriolosclerosis, I get all the hard words, and cerebral atherosclerosis. And she found that with higher exposure to PM2.5 in those 5 years prior to death, the risk of both of these atherosclerotic outcomes was higher. The prevalence was higher on autopsy. She also saw a weaker signal with respect to higher NOx exposure. Now, with respect to this other outcome, it's called cerebral amyloid angiography. There was not much going on, nothing that seemed consistent. And this isn't necessarily a contradiction of the results above, because this pathology develops through different mechanisms. Now, we're not the only ones in the room doing this. I want to also highlight some work underway led by Dr. Magali Blanco. She is here. You should see her poster. This is just a trailer. And so she too looked at these 3 outcomes and similar to us, found that higher exposure to PM2.5 in the ACT study in the 10 years before death did seem to be related or weakly associated with cerebral arteriolosclerosis and atherosclerosis, not so much with amyloid angiography. And yeah, I spelled that wrong. Angiopathy. So, by contrast, she did not find any association of NOx with these outcomes. And with that, I want to turn to, I want to turn to some recent developments on the exposure side. There were actually a lot, and it was hard to pick. So if you have interesting results on the exposure side, and you weren't included, it's not you, it's me. So back to Dr. Blanco. One other feature of her really cool research is that she's looking at ultrafine particles. And this is just the results with respect to some of the cerebrovascular pathologies. And where you can see she sees a suggestive association with cerebral atherosclerosis. She's also playing around with air pollution mixtures as well. And I want to turn away from the autopsy world for a second, get back to people and their clinical symptoms, and highlight the work of Dr. Boya Zhang. What she did is she estimated 10-year mean exposures to total PM2.5, as well as exposures to PM2.5 from 9 specific sources. And she did this in the health and retirement study for nearly 30,000 people living across the United States. And here's what she found, or some highlights. She found a lot. She found the expected adverse association of PM2.5 exposure with dementia risk. She also found that with higher exposure to PM2.5 from an agriculture source was associated with higher dementia risk. By contrast, with respect to the road traffic-related PM2.5 signal, that association, adverse association, was attenuated when she adjusted for the presence of other pollutant exposures, air pollutant exposures. And finally, she did see a rather troubling adverse association of a wildfire-based PM2.5 with dementia risk. And that brings me to wildfire. This is a nascent line of research. It's clearly important, for reasons I don't need to explain. And here's a sample of what we have so far. A study based out of Southern California. This is a study of over 1 million members of the Kaiser Permanente Southern California region. And what researchers reported was an adverse association of higher exposure to wildfire-based PM2.5 and dementia risk. This is a 3-year exposure. They also found this an adverse association with non-wildfire PM2.5. It was a much smaller in magnitude, although estimated more precisely. But by contrast, we have the results from Dr. Boya Zhang in a location or a setting in India, where what she reported was that exposure to this pollutant was not associated with cognitive performance and actually associated with slower rate of cognitive decline. Now, there are reasons that kind of counterintuitive result can happen, but clearly we have a lot more work to do. And with that, I will turn things over to Dr. Corey Slechta.
Well, good morning, and thank you very much for the invitation to be here. We're going to talk a little bit about what's going on in the world of animal studies and air pollution, and I'm going to stick mostly to developmental exposures. There we go. So you've heard there is, and I'm sure everybody in this room knows, there has been this accumulating evidence linking air pollution exposures not only to neurodegenerative diseases and disorders, but also to neurodevelopmental disorders. And this list keeps growing, which has led some people to say, you know, is this something spurious? How can air pollution be associated with all these different neurodevelopmental and neurodegenerative kinds of conditions? And there's probably two reasons for that. One is that there is probably a commonality of some of the contaminants in air pollution, which of course is a mixture of contaminants, that occurs across geographical areas and studies. And in fact, in those studies where maybe you don't see effects, maybe some of those more important contaminants that lead to the neurotoxicity aren't prevalent in the mixture. But secondly, and something I don't think we think enough about, is that we talk about these neurodegenerative diseases and neurodevelopmental disorders as unique entities, but the reality is they share an awful lot of features in common, and it's very likely that air pollution is acting on those features that they share in common. And this does not, oh, there we go. This is just a table showing you some of the features that are shared in common across all these neurodevelopmental diseases and disorders. One, which I'll talk more about in a minute, is called ventriculomegaly. It's in large lateral ventricles in your brain through which the CSF, the cerebrospinal fluid, flows. You get alterations in the myelin of the brain, and therefore you can get disconnection of your two hemispheres. Too much glutamatergic neurotransmission, what we call excitotoxicity, neuronal cell death, inflammation, microglial activation, oxidative stress, and a host of behavioral deficits as well. So these are all shared, suggesting that air pollution is probably acting on those shared features. Okay, so that raises a lot of questions because air pollution is a lifetime exposure and begins obviously during development. So some of the questions that you can think about is what is the trajectory of air pollution impacts on brain, particularly the frontal and striatal systems that underlie a lot of the behavioral deficits that we see after there's been a developmental exposure. Does air pollution cause reprogramming of these systems? And if so, that's something that's permanent. Do alterations at different physiological stages, for example, adolescence or aging, basically trigger recurring adverse effects? And one that I'll show some data on is behavioral experience. Once you and I are born, we engage in behavioral experience across our life. It has requisite for significant brain activity. And so can air pollution modify that as well? Okay, so I'm going to talk about ultrafine particles, which we've heard a little bit about. They are considered the most reactive component of air pollution. And the picture on the left shows you the size of these. You heard a little bit about this yesterday. Ultrafine particles are not regulated by EPA, and at least some of the data that's out there now has said that reducing levels of PM 2.5 does not necessarily reduce levels of ultrafine particles in the air. So the ultrafine particle with a carbon base has more surface area per mass and therefore can contain more contaminants than the larger particles. And these can include organic constituents as well as metals, in particular redox active metals, which I'll talk more about as we go forward here. So let me talk a little bit first about some studies in mice. I hope you're not big fans of mice. Most people are until they show up in their kitchen or their basement. But I want to talk a little bit about postnatal exposure of mice. This is exposure that occurs right after birth, and in most mouse species, that is equivalent to the human third trimester of brain development. So I'm going to show you a little bit of data from studies in which mice were exposed on postnatal days 4 through 7 and 10 to 13. These are exposures for 4 hours a day. You can see on the left our facility in which we're pulling air in from outside, separating it by particle size, concentrating the ultrafine component, and then delivering that into a whole body inhalation chamber. So these are ambient inhalation exposures that we're talking about. We take brains from these animals at different time points, and they also undergo behavioral experience. So I mentioned ventriculomegaly, and if you see at the very top left there, you'll see what the ventricles of the brain look like at postnatal day 4. So early in the period of brain development, your ventricles are very large. However, as the brain continues to develop, neurogenesis goes on, gliogenesis, the lateral ventricles tend to shrink down, and I think you can see in the top right, I don't know if I have a pointer here, where it says LV is what a normal lateral ventricle should look like at postnatal day 14 in a mouse. On the bottom, what you see are brains from male mice that were exposed to the ultrafine particle component at the same time. I think you can see the enlarged lateral ventricles that you can see across 3 different animals there. On top of that lateral ventricle is something called the corpus callosum. That is the largest myelinated tract in the brain that runs all the way rostral to caudal and basically connects your 2 hemispheres. And it literally looks like Swiss cheese. We've seen these effects now down to exposures of 22 micrograms per cubic meter. Haven't been able to get exposures lower than that as of yet. Are these permanent effects? Yeah, they're permanent effects. You can see on the right, brains from males taken at postnatal day 270, which is well into the lifetime of a mouse. You can see that ventriculomegaly is still there. And that's largely because all of the things that should have reduced, the gliogenesis and the neurogenesis hasn't occurred, and those time points are now behind. There's no way to make that up. Okay. In addition to that, as I mentioned, the corpus callosum, the largest white matter tract of the brain right above the ventricle, is reduced in size in males. There's a male bias to this effect, and you can see that on the left, something that we don't see in females, at least at this early time point. Now, one of the things I don't have time to show you today is that a lot of effects in females aren't showing up early, they're showing up later on with these developmental kinds of exposures. The right side shows you myelination of the corpus callosum. You can see in an air brain, that orange area is myelin staining. About 90% of the corpus callosum is comprised of myelin. And then you can see in the CAPS photograph the loss of myelin. Not only is the corpus callosum reduced in size, but the amount of myelin that's left is very, very reduced in males. Again, a male biased effect, which is particularly interesting given the male bias of a lot of these neurodevelopmental disorders. We also can see at postnatal day 14 cell death. You can see this on the left side where the arrow is. These are loss of neurons in what's called the nucleus accumbens. That is a very important dopaminergic region of the brain. It mediates learning and cognitive and executive kinds of functions. And you can also see in the males an increase in what's called oxidized glutathione, which is the brain's antioxidant, suggesting that there's redox activity that has been going on in these exposures. Another effect that we see is excitotoxicity. The brain is producing too much glutamate. Glutamate is an excitatory neurotransmitter. It is usually countered by GABA, the inhibitory neurotransmitter. You can see at postnatal day 14, there's a trend in both sexes to have an increase in excitotoxicity. This effect actually gets worse in males as we go out to postnatal day 60. I'm going to show you a little bit of behavioral data. We see a lot of different behavioral phenotypes here, and we tend to focus on behaviors that we can look at in both human and animals using the same paradigms. This is one that we measure impulsive-like behavior, and we have a mouse in a Skinner box or operant chamber, has to press a lever to get food reward. This mouse has to make 50 responses to get the first food reward, and then it can go into a period where if you just do nothing at all, you keep getting free rewards. The only trick here is that the time between the free rewards keeps getting longer and longer. And so if the animal makes a response, they have to start the 50 responses over again. So we're looking at their ability, their impulsive-like behavior. And I think this is, This is what we call a cumulative record of the animal's behavior under these conditions. On the top, you're looking at an air animal, and we're looking at cumulative responses on the y-axis and session time. On the x-axis, you can see the air animal makes 50 responses, and then, as you'd expect, starts waiting and getting free rewards. Eventually, it gets too long, and the animal starts the ratio of 50 responses over again. I think it's quite clear. You can see on the bottom the caps exposed males cannot wait, do not wait under these conditions whatsoever. And in fact, what we see quantitatively is a decrease in their mean wait time and an increase in the number of times they start the ratio over again. In some of these experiments, we also re-exposed animals as adults to caps, and that made the mean wait time even shorter, so increased the impairment, if you will. So one of the things that we've been thinking a lot about is what are the components of air pollution that lead to neurotoxicity? And one of the things, of course, that's in these are metals, including redox active metals on those ultrafine particles. And these ultrafine particles have direct nose-to-brain transport. They are, all your life, you are breathing them in. They are going up the nose, traveling on axons directly into brain. So they bypass the blood-brain barrier. And consequently, when we're measuring levels of metals or things in serum, they may not actually reflect what's going on in the brain because this is all bypassed that blood-brain barrier and its communication with the periphery. So what are some of the metals that are in air pollution? Well, the top three are iron, aluminum, and silica. Remember these because I'm going to show you some micrographs here in a minute. And the thing about those is that iron, which is one of the most prominent, is not just something that we see in the US, we see it around the world. Iron is a redox active metal, which means it can cause oxidative stress. And you're probably thinking, yeah, but our brain requires iron. But it's like anything else. Too little is bad and too much is bad. You are actually accumulating iron in your brain across the lifespan. We know it's elevated in aging. And it's elevated to an even greater extent in every neurodegenerative disease. There is an increase in brain iron. And the brain doesn't seem to have any good mechanisms to eliminate iron. It probably didn't need to before the Industrial Revolution became something. So this is just some laser ablation ICP-MS images of animals that have been exposed to air pollution. And you can see on the left the levels of iron and sulfur after these exposures are hugely, dramatically increased. The top right shows you aluminum, and I'm still just boggled by this picture because I don't know if you can see it, but the aluminum literally is ringing the hippocampus, very high levels in hippocampus. But also increases in levels of calcium and copper. And this seems to come at the expense of manganese and zinc, which are also required by the brain. So there clearly is a brain metal dyshomeostasis that goes on with these exposures. Here we're looking at some particle deposition in corpus callosum. You can see particles of iron, silica, and even agglomerates of iron, silica, and aluminum. Clearly, aluminum and silica don't belong there at all. So one of the things we've started to look at are nanoparticle iron inhalations in mouse, and we've done these to iron nanoparticles alone or iron nanoparticles in conjunction with sulfur dioxide. Why in conjunction with sulfur dioxide? It was shown back in 1980 by Joe Brain's group that sulfur dioxide actually increases the uptake of iron, and it also affects its distribution. And as you'll see in some of these data, that speciation makes a big difference. And so iron can also cause something called ferroptosis, which is a form of cell death that's different from other forms of cell death. And it's characterized by iron accumulation and lipid peroxidation. And it affects what's called the transsulfuration pathway, which is how you produce glutathione. So what happens when we do these exposures? Well, iron with sulfur dioxide results in both female and male ventriculomegaly, which is significant in the males and not in the females, so reproduces characteristics of air pollution exposure. What about its effects on brain neurochemistry, particularly this frontostriatal circuitry? And I want to show you what happens, after behavioral experience. So as I said before, when any of us engage in behavioral experience, our brain neurotransmitter systems go essentially into overdrive. They're working, producing neurotransmitters. Those neurotransmitters and neurons are talking to each other. And here what we're looking at are changes in the brain neurotransmitter levels after that behavioral experience, looking at glutamate, serotonin, and dopamine systems. And I think if you just focus on the bottom, which is males, you can see that in the males that were exposed to iron and sulfur dioxide, they can't increase their brain neurotransmitter levels in any of those systems to that which is requisite for undergoing behavioral experience. If we look at iron alone without sulfur dioxide, now we can see there's effects in both males and females, and it's primarily in the striatum in this case, and resulting in overshoots of neurotransmitters, so excessive dopaminergic as well as serotonergic function. So speciation makes a big difference in terms of brain effects. Finally, I want to talk a little bit in my last couple minutes about something that we've now been seeing, which is mind-boggling to say the least. We are finding in the brain these things called corpora amylaceae, CAs, and they were actually first reported by Purkinje back in 1837 and have since been totally ignored. Up until about 10 years ago, people started looking at them and saying, oh, we think these are collecting debris in the nervous system, so they're calling them wastiosomes. They are polyglucosan bodies with no membrane, and you will tend to find them near the vasculature in the brain. What we're finding with these is that iron that comes in through the nose directly into the olfactory bulb, which is where these are, that's the portal of entry into the brain, are being taken up by these CAs. And we know from other studies that I'm not going to show you right now that this is in fact the exogenous magnetite that we also see on the filters. At the same time that the CAs are taking up this iron, they are inducing ferritin, which is a regulator of iron. So we're finding both iron and ferritin inside of these CA bodies. These are some data that Jennifer already talked a little bit about this study from the Rush study. We see exactly the same thing in human brains. This is from the Rush cohort. You will find these CAs in human brains. They also contain both magnetite and the ferritin. And we're looking as well at other metals that are in these, potentially in these. Two things that I think are really mind-boggling about this. What you're looking at here is the inside of a neuron, And inside of that neuron, you can see these CAs, and it's literally carrying aluminum and silica, raising questions, at least in my mind, about whether these CAs are involved in metal balance, ionic balance in the brain. Something that we're working to, again, to look at. And secondly, one of the things that you see in air pollution, at least we do in specifically in mouse brains, the demyelination that you see is actually a vacuolization. So you'll see holes in the myelin. And one of the things that we've now seen here is a picture of myelin that is breaking open, and inside that breaking open myelin are CAs, suggesting to me that accumulation of these CAs, which is probably increasing with the metal content from air pollution, are breaking open that myelin, and so is this a new mechanism of demyelination that we hadn't thought about. We have seen pictures of corpora amylacea that agglomerate at the vasculature, and that agglomeration causes a buildup such that they break down and re-release the contaminants, the iron, and whatever else is in there. So, to be continued. Let me just end by saying that exposures to real-time, real-world, human-relevant levels of ultrafine particles, which we are not regulating, during periods of brain development can produce phenotypic characteristics of both neurodevelopmental disorders and neurodegenerative diseases. These corpora amylacea, just mind-boggling, they were first described in 1837, and when I talk to neuropathologists, they go, oh yeah, whatever. May play a role in ionic balance and in vacuolation. And we need to spend time thinking about the components of air pollution, because if we can identify those that are the most problematic, maybe we can do a better job of protecting public health. So let me end by saying it really does take a village, and I want to in particular point out the role of Günter Oberdorster, who I'm sure most of you know, and Ushi Graham in these studies. Thank you.
Thank you. And so we're going to move to questions, and I'll take again the prerogative of helping set this up to ask a really simple question of all of our speakers. So thinking holistically with the potential role of PM 2.5, we have multiple health effects, cardiovascular, respiratory, neuro. So is parsing it out by discipline the best approach to take, or should we start to try to think more holistically about this being some common denominators that either inflammatory, vascular, or others that may have different manifestations depending upon individual susceptibility? And if you could solve that question in the next minute, that'd be great.
Well, I definitely believe that's the case, that we need to be looking more holistically. As was said before, the brain is connected to the rest of the body, and the world has finally begun to recognize that, and so to me that makes the most sense. We typically do it with these studies by sharing tissue and that kind of thing, so at least it's a way to start that.
Yeah, it's funny because I think about how do I get money, and don't get money that way. But the reality, as you could see in both the work that Deborah was, basically all of us, that if we if we focus too narrowly, we end up missing a really important part of the signal that matters, and our job then ends up being to translate those signals to human health, and people really do care about this. I will just say, anecdotally, we have said forever how people can, we've told people about the adverse effects of air pollution. When you start telling them that air pollution is going to affect their brain, they listen a lot better.
Yes, I agree with everything Deborah and Jennifer said, and I think it's important when we think about the data we're creating, how do we use it to translated into action and change, and thinking about solutions, then we have to really think about holistically what's happening at the population levels. How can we support communities, but how can we support individuals and nations and so on. So when we start thinking about broader and broader, we have to really bring in our epidemiologist colleagues with neuroscientists, with healthcare physicians, even nephrologists, pediatricians, neurologists, psychiatrists, environmental health specialists, public health specialists. We all have to come together.
Okay, I'm assuming that folks are not just holding up a wall, but are waiting for questions, so go ahead and jump in.
Hi, Meredith Franklin, University of Toronto. I have a question about exposure windows. So this may be a question for Dr. Weave, a little bit more for dementia, and was struck by the exposure windows for Alzheimer's or for dementia were, looking at 10 years or 1 year before death. That seems like too short. And I was wondering, why are those time periods used, and is that correct?
Well, thank you for that question. I think it's plausible to assume that air pollutant exposure is probably affecting multiple parts of the development of dementia, including its pathology. I too am skeptical about that 1 year window, especially prior to death. And so with respect to anything that involves neuropathology, I think we need to take a step back and try to expand the options for windows to evaluate. In sympathy, because I'm doing some of this too, with the folks doing autopsy research, often you don't have a lot of choices because you don't know where people lived, and we're trying to make progress in that regard. But moving forward, I get a sense in the field with my other colleagues who are doing this, which is this effort to try to capture more windows in the life course.
Great. Thank you.
Thanks very much, Michael Brower from UBC and IHME. I'm actually going to riff off Meredith's question. So I want to ask actually about window, but also patterns of exposure. And we seem to sort of have we have some evidence actually from wildfires and even some studies that have looked at long-term exposures and residential histories, that it is that sort of proximal exposure that gives you the greatest signal. On the other hand, there's this sort of concept of, neurodegenerative life course. So I'm just wondering if maybe like evidence from CTE, completely different sort of cause where you have, events that sort of exceed a threshold over a lifetime. And we've sort of been obsessed with average exposure, but is it time to maybe think about patterns, peaks, intensity, something like that? Just like to get your thoughts on that.
You want to talk about like what you see in mice?
Yeah, it's really, it's interesting in the studies that we do, we can't control what the concentration of air pollution is on any given day. So doing direct replications is hard. That's why I've tried to structure essentially concentration effect curves and see where we are. But we don't have enough data yet to really be able to say, are there peaks, are there, and we don't have enough data on speciation at those different times yet to be able to say that either. So, but I do think that's an interesting question. Is it the peak of a high day versus consistent exposure?
I just want to point out about something. In the field of neuroscience, neurodegenerative diseases are really considered a nightmare to be able to find solutions, because by the time someone goes to a clinic or see a doctor and they get diagnosed with these diseases, because their symptoms of memory loss or movement disorder are coming to a stage of really impacting their lives, and they're like, okay, so I want to get it checked out, it's actually too late in the disease process. We know that more than 50% of the neurons at that time are usually lost, and the disease may have started years, sometimes decades earlier. We see that with a lot of TBI, or even having a car accident hitting their head, and then these people can have the neurodegenerative disease following 5-10 years after that. And so I think just resonating from my colleague said, it's really important to look at life course and what we, and even thinking about, like Deborah had mentioned at the beginning of her talk, neurodevelopmental, that stage actually gives us, when the babies are born with the double number of neurons, neuronal connections they have, and they lose half of it over the first 5 years of life, refining those connections. And those are really critical period of loss and regeneration that we have to study to learn more about how the external factors may be impacting that neuronal loss and regeneration. So those are really critical stages to look at, I think, to learn about even neurodegeneration.
Well, there is this study led by Christina Dung. So what she did is she conducted what's called a sequence analysis, and involved in that was the identification of temporal patterns in, I believe, it's 3 pollutants. So PM2.5, NO2, and what was the other one? Ozone. Thank you. So, and then identified sort of the most common patterns. So to give you an example, like high PM2.5 for the entire 10-year period, plus high NO2, plus high ozone, for example, and various patterns of decreasing and increasing pollutants. And overall, her findings suggest that the worst scenario is to have this consistently high exposure. It did seem that people who had decreasing exposures over time did experience benefits from that. I will say that this is where we want to go. We want to know, like, can we do anything? Like, is it ever too late? And for CTE, we're not. I will say I'm familiar with this because I worked with this group. It's a different story. But I want to just leave you with this challenge. When we look at changes in exposures over time, the reasons those changes can happen are because of changes at where they're living. So just pollutants are going down. The other reason that those exposures can change is that people move. And we have to be super careful in understanding the reasons people are moving. Often people are moving because of their health. Often they're staying because of their health. And so it doesn't mean we shouldn't be asking these questions, but it's something we have to be thinking about.
Hi, Chris Carlson, University of British Columbia. You distracted me by your last comment. I don't know how much data there is that people actually move because I'm sure some people move because of their health, but I think it's kind of unusual, relatively, but that's maybe for a later discussion. Kind of a detailed question, but it was mentioned by 2 of the speakers, the first and the last, the olfactory bulb, and I'm afraid to ask this question when Gunter's staring at me, but but nonetheless, there's been a study recently, relatively last couple of years, from Manchester in the UK that was a controlled human exposure, and that's kind of my interest and expertise. And they applied nose clips and found that it didn't change the signal whatsoever. So again, apologies for those that aren't as interested in mechanisms, but I found that really intriguing, and I found it at least somewhat questions the nasal route. And I'm just wondering, since it was mentioned by 2 of you, what you think.
The trigeminal nerve.
Yeah, I mean, it isn't, you can get those particles traveling on axons. There's the trigeminal nerve. You can even get in from the eye. And when you have a nasal clip, I don't know that completely blocks the inhalation via the nasal route of exposure. But I think it's pretty well known that particles can travel on these nerves directly into the brain.
Yeah. Maybe, I'll talk to you about maybe I'll ask. The last part that you said about the metals dissociating kind of proximal to the CNS, or the brain really, is that right? You said you're showing the image of the carbon core and then the metals dissociating. Is that something that the dissociation specifically at that near-brain interface? Is that something that's speculative or known?
Well, people have actually been looking at, there are protein coronas that can form on these particles, and they've shown that they basically were focused on the organic contaminants, but as the particle travels, the nature of that protein corona changes, and there is dissolution of the metals once they get into the olfactory bulb itself and can even travel further into other regions of the brain. So there's a lot of chemistry of those particles actually going on once they reach the brain.
Thank you. Yeah, I really appreciated your talks. Thank you.
Hi.
So if you can bear with me for a second, I think one other thing, just as a recollection, for a lot of the neurodegenerative diseases, changes in olfaction is oftentimes an early warning sign. It's also, as I always tell folks, because I used to deal with nose-to-brain transport, it's also one of the few portions of our nervous system in direct contact with the environment. And so that's an area of research that I think is not being as explored as heavily as we might want to look at, because they are a susceptible population of neurons there. Yes?
Yeah. Hi, Jim John from Duke University. This is for, actually, my question was, part of the question was already sort of asked just previously to Dr. Debbie Carr-Slechta about the ultrafine particle, the exposure, the distribution kind of thing. So thank you for the previous question. The follow-up sort of question is, for someone like me sort of doing the exposure health effects thing, right? So your Aaron kind of finding, that's what's fascinating, because we, did particle composition metals 26 or something. Never thought about really look at iron, because such an abundant element at a good side. But again, my studies in the past was looking into lung and cardiovascular, so we'll not see anything like that. But now reminded about the sort of brain health effects for if I were going to do a study in the real world, in epidemiological setting, what would you advise to do it? Because I assume inhale into the lung, and then the iron going to accumulate in the brain. Can you tell a little bit more about how to sort of get that sort of determined? Because I don't know how much goes to where. Apparently that distribution will make a difference.
I don't know that we know the answer to that. We haven't really looked at what's happening so much in the peripheral system. Other people, as I mentioned before, have reported that brain iron is not related to what you're measuring in the periphery. But yeah, that's why your question is why we started going to individual components to try to take a look at those kind of things. But it goes back to the original question that Dave brought up, which is we need to look more at the entire physiology and trying to determine what's actually happening here. I don't know if that answers your question.
Yeah, I'll add that it's so extraordinarily difficult to do, and often we're left with snapshots of people. For example, using autopsy, we do have a newly funded study using autopsy data from Brazil, where most people do undergo autopsy at death. And so we are looking at a range of tissue, including lung and brain, but it's still a snapshot. It's still what was there at death. So lots of innovation opportunities here.
Hello, and thank you for the great presentations. I'm Bonnie Holmes-Jen. I'm with the Air Resources Board in California. And it was mentioned early on that the most recent ISA had found this association for neurodegenerative illness at, I think, likely causal. And there's also, I believe it had a finding of suggestive of causality for birth outcomes. So there's been a lot of evidence, and then all these recent studies that you've been showing. And we just completed a study also on birth outcomes and neurodevelopment, and definitely saw increased associations with PM2.5 for preterm birth, terminal birth weight, and Alzheimer's disease. So I guess I'm just leading up to, given all this research, what do you think are the outcomes with the most robust evidence of effects that could be used in regulatory analysis? Because we know that reducing levels of PM2.5 does reduce exposure, and we want to see these brain health. We want to educate the public more about the brain health benefits and make these connections that you're talking about. So, and I mean, even though these findings were in the ISA, the EPA, nationally there has not been a regulatory analysis of these benefits, but we are looking at how we could do this analysis in California. In fact, there is a piece of it, I think, I guess there is some look at nationally at the benefits from reduced hospitalizations for Alzheimer's. But which outcomes do you think are most robust? Thank you for listening to my long question.
Excellent question. I would love to talk after too about your projects in California, because we're also doing some projects in California, also in India and other places. But I think the most evidence we have right now is around dementia and neurodegeneration more than birth outcomes and also neurodevelopment of children. That being said, that data is coming now. We are doing a systematic review right now looking at exactly that prenatal exposure to birth outcomes and then the first 5 years of neurodevelopmental outcomes in children from the prenatal exposure. And so the study is going, but I can share what we have so far. And I think also but I think that's going to be even by itself, the neurodevelopmental part is a really critical when we think about policy regulations acting on bringing, and perhaps, again, dementia could be brain health across the lifespan. And we're also doing some studies looking at children's cognition, at school-age children's cognition and mental health in India, in New Delhi, due to air pollution and heat together, exposure to those. And also mental health is coming really into focus now as well. So those are some of the elements that we need to bring more data in, but those will be really critical in our fight for action through the policies as well. Yes.
So I'm not going to add, I'm just going to say, based on my watch, I think we have like 20 seconds for a really quick question and response. So I don't, I'm just trying to be aware of time here because we don't have a short break coming up. But
great. Thank you. Magali Blanco from the University of Washington. I'm wondering if you could speak on life course exposures and how we might start to tackle these in this field. It seems like there are two pretty big problems with when we're thinking about human studies, and one is just the data that are available, and the second are the methods, and how do we actually tackle these? Thank you.
I really think mice is where it's at. I mean, there's such tiny little lives, except for the ones that are in our houses. So, yeah, there's some, just generally speaking, methods and life course exposures. And maybe, Sarah, you can add some things from like what's really happening in the GECC. So we can think about methods such as synthetic cohorts. These are essentially cohorts that are assembled from different age groups to form one long lifespan. And if we are able to get exposure data from each of those stages and meet some pretty strict assumptions, that might be one way to start looking at life course. And I think, we just have to acknowledge that it's not going to be perfect in all these sorts of potential solutions. There is also the possibility of following cohorts forward. And so instead of, there's been a history in dementia research of recruiting cohorts who are 65 or older. And we know, everyone agrees, by then it's too late. But actually it's not too late, but it's too late for these earlier life questions. And so we can take advantage of cohorts that were recruited at much younger ages and follow them up. And that's been happening to a great degree just in general for dementia development and dementia research. There is also the possibility of trying to reconstruct people's address histories, and I've been there. And it is way harder than you think it's going to be. And so this is still an opportunity for leveraging actually mostly available information to our benefit, but first determining how accurate it is. So I should say before I turn this over, even if we can get those histories, we still need the exposure. So if I can get someone's exposures back to the 60s, or their address back to the 60s, I may not have very good measures of their exposures. I might be able to guess, though. So it's the holy grail, Magali. Yeah.
Okay, so I want to thank Sara and the speakers for putting the session together. I think it was very informative.